very good morning, students. So today we are with one another topic, which is the functional evolution of the brain. Now this is a very important topic as per your examination is concerned. So before starting with this particular topic, I should give you a brief idea that what we have to do in this particular topic. Actually, we have to correlate the evolution of the anatomical aspects of the brain with the functional aspects. So if we go from like fishes, if we go with the fishes up to the mammals, so what we have to see is that what is the anatomy of the brain and how the functions they have changed with the anatomy of the brain. Now suppose if I take one example, suppose in the case of the birds, we have a very good eyesight. So if there is a good eyesight, the scientists they have seen that the brain of the birds, they have got larger optic lobes. So that means if you have larger optic lobes in the brain, you can have a good eyesight. So this is a kind of correlation of anatomy with the functions. Now if I talk about the say brain of the dogs, so dogs they have a very good smelling power. So what they have is that they have got larger olfactory lobes. So if you have larger olfactory lobes, you can have a better smelling power than the other animals. So actually what we are doing is that we are correlating the anatomy with the functions that the animals perform, or what the extent of the senses they have got in relation to the anatomy. So actually this is the functional evolution of the brain. So if we start with the fishes, suppose we take the brain of the fishes, it is bit simpler in the structure, but as we go above the level of the fishes, if we go to up to the mammals, the complexity of the brain, the anatomical complexity, it increases class by class. If we go with amphibians, they have got peculiar brains. If we got reptiles, we have got more complex brains. If you have got say birds, they have got different anatomical aspects of the brain. Still the parts which are like present in the brain of all these animals, they are similar, but the different parts of the brain can have different sizes. So if they have different sizes, they have got different anatomy. That means suppose birds, I give you one more example, that the brain of the birds, they have got very less developed cerebellum. So it is an anatomical feature. So the birds, they have got less developed cerebellum. So this is a part of anatomy of birds. So that can refer to like less functions of equilibrium that are concerned with the cerebellum. So overall, what is this particular topic all about is that we have to like go with the evolution of the brain anatomically, and then we have to see the functional evolution also, that how the functions of the brain they have changed according to the anatomical features. So this is the actual topic what we have to do in this particular topic. So now let's start with the functional evolution of the brain. So I have divided this particular topic into 4 subparts. So I have taken some subparts. The first part which I have taken is the medulla. So I'll be taking the medulla in the very first part of this topic. The second part that I have taken is the cerebellum. So the third part that I have taken is it is the midbrain. So I'll be talking the talking about the functional evolution of the midbrain in the third part, and the fourth part that I've taken is the forebrain. So basically I have divided the functional evolution of the brain into these four parts. Now let us start with the very first part, which is the medulla. So students, the medulla, it is a very important site for sensing. So this is the important part. So this is very important in the sensing process. The medulla, it is actually the part which is responsible, you can say, for the you can say the originating point. It is the originating point of the cranial nerves. So the medulla, it serves as the point of entrance and exit of the cranial nerves. So this is a very important function, you can say, of the medulla. So it also provides a way station kind of a situation. It is a way station for their fibers into the, you can say, the brain. So the most important thing which the medulla has, it is the respiratory center. So the respiratory center, it lies in the medulla. So the respiration, it is under the control of the medulla of the brain. Now we have to see that how this respiratory center, it is like acting in different classes of, you can say, the animals. If we have fishes, amphibians, reptiles, birds and mammals, that how these respiratory centers, they function in the respiration, and what is the anatomical and physiological, you can say, evolution throughout the course of the journey from fishes up to mammals. So now I am taking the very first part in the medulla, which is the medulla as the respiratory center. Now first of all, I talk about the mammals. So in the case of mammals, the respiratory center in the medulla, it lies actually in the mid-dorsal part of the medulla. And if I have to give you a diagram, suppose this is the medulla part, and where is the exact position of the respiratory center? It will be in the mid-dorsal part of the medulla. So in the midline on the dorsal side, we have the respiratory center. So actually this is the respiratory center. So we can say this is mid-dorsal in position. So this will be the medulla, and we have the respiratory center in the mid-dorsal position. So this is the case of the mammals. And if I talk about like cats, monkeys and dogs, so here in these animals, scientists, they have found that the respiratory center is further divided into two parts. So it is divided just like this into two parts. So the upper part, it is the expiratory center. So we can say that this is the expiratory center, which is the upper part, and the lower part is the inspiratory center. So both these inspiratory and expiratory centers, they work together to form the actual respiratory center in the case of the mammals. So as we know that we have phrenic nerves which are coming down up to the intercostal muscles of the chest, which are expanding the chest, and they are contracting the chest. So when the inspiratory respiratory center, it gives impulses to the phrenic nerves, what happens that the chest expands and the air flows into the lungs. And whenever the expiratory center, it gives impulses, what happens, that the phrenic nerves, they will help to contract the chest and the air will flow out of the lungs. So this is the typical you can say respiratory circle in the case of the mammals. So what I'm like concerned here in this particular slide is that medulla has got the respiratory center. It has got two, you can say, further differentiations of inspiratory and expiratory centers, and they both are like working together as a respiratory center in the case of the cats, monkeys and dogs. Now after mammals, now we are talking about the turtles. Now in turtles, the breathing is apneustic. Apneustic refers to the prolonged inspiration. So they can take air for longer times in this apneustic kind of inspiration. So in the case of the turtles also, there is an evidence of the inspiratory and expiratory centers as in the case of the mammals. So as we have taken in the previous case that we have got two further differentiations of the respiratory center, turtles, they also have got these two respiratory centers. Now students, we have the case of the frogs. Now in the case of frogs, it has been seen that if the vagus nerves, or you can say the vagi, if they are stimulated, so what happens that these are excitatory to the lung musculature, and what has been seen that here the vagi, they are inhibiting the spontaneous lung contractions during the breathing. So if in frogs the vagus nerve, it is stimulated by some impulses, so that will inhibit the lung contraction. So it has been seen by various scientists. Now if I talk about generally the reptiles, now here I should take some examples. I am taking just an example of turtle and snake. So in these two cases, what has been seen that the vagi, if they are stimulated, on the contrary, these vagi, they are excitatory to the lung musculature. So in the previous case of the frogs, we have seen that the vagus stimulation, that is inhibiting the lung contraction, but here in the case of the turtles and the snakes, we have seen that the vagus stimulation, it is excitatory, or it is you can say enhancing the lung contractions. So these contractions, they can be like you can say controlled by a combination of the vagi and the respiratory centers. Now if you have like more of the stimulation of the vagi in the case of turtles and snakes, you can have more of the lung contractions, and lung contraction can be like expiratory in function. So when the lung will contract, what will happen? The air will come out of the lungs. So that means it is expiratory in nature. So if you have like less of the vagi stimulated, it is inspiratory. If the vagi are stimulated, that will cause the expirations. So here in the case of the reptiles, especially in the case of the turtles and the snakes, so there is a like a bit combination of the vagi and the respiratory centers that help in the breathing process. Now, after many experiments, scientists have discovered a new center in these two cases. Now, in the case of turtles and snakes, so what they have seen is that there is a center which is like giving impulses to the vagi. Now, this particular center, it is giving a negative kind of you can say stimulation to the vagi. So what this center is doing, it is actually inhibiting the vagus nerve. So whenever this particular center, it will inhibit the vagus nerve, what will happen, that will give a negative impulse to the vagi. Now vagi will not be stimulated. There is no stimulation as such in this particular case. So there will be no lung contractions, there will be no expirations. So that means more of the inspiratory, you can say, impulses will be given to the lungs, more of the inspiration can take place. So that means this particular center, along with the stimulation of the vagi and the respiratory center, which is the actual center which is controlling the respiration, these three systems, they are actually controlling the respiration process in the case of the turtles and the snakes. Now students, we enter into the second part, sub-part of the medulla, and now we are taking up the medulla and the autonomic functions. Now how the medulla part, it is taking part in the autonomic responses by the like different animals, and how this evolution occurs from fishes up to the mammals. So first of all, the medulla in the case of mammals, it is, if I take the case of mammals, it is containing a vasomotor center, and scientists, they have found that this vasomotor center, it is actually causing the vasoconstriction actions. So I'm just writing VC. So this is vasoconstrictions. So whenever you have like low of the blood pressures, in the case of the low sodium concentrations, so you can have a higher vasoconstriction value and you have more of the blood pressures. So that is a kind of, you can say, homeostatic mechanisms that is found in the mammals. So here in mammals we have got a vasomotor center in the medulla part, and this vasomotor center has a vasoconstriction effect. Now, after like various experiments done on the mammals, scientists, they have also found that there are some vasodilation cells also which are present in the vasomotor center, which are actually vasodilating in nature. So that means these two types of cells, which are vasoconstrictors and the vasodilators, they are the part of the vasomotor center. So now we have found that both these types of cells are present in the vasomotor center in the case of the mammals, and this center is directly related with the medulla. So that's how this is a kind of autonomic function which is related to the medulla part. Now, after the vasomotor center, there are some other functions which are involved like with the medulla, which are under the autonomic functions. So the first is salivation. So the salivation function is under the control of the medulla. Second is the vomiting. So the phenomenon of vomiting is also under the autonomic functions, which are done with the help of the medulla. Next is very, very important function, which is called as the postural control. Now if we stand like straight, so our posture is straight, so this is because of the autonomic function of the medulla. This is a very important function. I will be discussing this postural control in a more elaborated way. Next is the equilibrium. So this is again an autonomic function which has got a relation with the medulla. So these are some of the functions. Again, we have the vasomotor center. So these are the 5 main autonomic functions of the medulla that you have to give in your examinations. Now students, there was an experiment done, and what we have done in this particular experiment, that we have taken a dogfish, and scientists, they have cut one part of the medulla of this fish. So half of the medulla was cut. So what happened, that what has been seen in that particular fish, it is showing spiral movements. So the fish will be moving spirally into that particular direction, uh, like in which the half of the medulla has been cut. Suppose the left half of the medulla has been taken out from the fish, it will be moving spirally to the left side. So that means the half that we have cut, left or right, it will be moving spirally into that particular direction. The eyes, they also deviate toward the operated side. So the eyes of the dogfish will be towards the left side. So this is the actual, you can say, the postural function of the medulla, which has been shown by this particular experiment. So we have lost the equilibrium in the case of the dogfish with the cut medulla. We have lost the postural control in this particular case also. So this experiment proves that the medulla, it is very important for the postural and the equilibrium controls in the case of the fishes. Now students, I am giving you one more example of the autonomic functions, and this is the Mothner cells of the goldfish. So these are very important cells in the case of the nervous system of the goldfish. They are associated with the medulla part. They are associated with the functions of the medulla. So what has been seen in these Mothner cells, that when these Mothner cells, they are destroyed in the goldfish. Now if these cells, suppose they are destroyed, so there are actually two Mothner cells present in the goldfish. They are very large sized cells which are present anterior to the optic lobes of the goldfish. So when they are destroyed, what has been seen by the scientists, that after the destruction of these Mothner cells, there is no change in the equilibrium, or there is no change in the swimming movements, because we have an intact medulla in this particular case. We have not destroyed the medulla part. Actually, we have destroyed the Mothner cells of the goldfish, which are actually very closely resembling or closely associated with the medulla. So there is no change in the swimming pattern, there is no change in the equilibrium in this particular case, but there is a tendency for early exhaustion during the maintained activity. So there is lethargy, there is inefficiency in maintained work. The fish cannot swim for longer times. It will be lethargic, it will be passive. So exhaustion that will take place if the Mothner cells are destroyed in the case of the goldfish. So actually Mothner cells, they are not directly controlling the postural control in the goldfish, but they are associated with it. So this particular experiment, it is giving the indirect relation in between the postural control and the Mothner cells of the goldfish. Now we have intact medulla in this particular case. We have not destroyed any part of the medulla. We have only destroyed the Mothner cells, which have got some extent of postural control, which is shown by the inefficiency in the maintained swimming actions of the goldfish. Now students, I am just talking in general about the fishes and the amphibians. Now in this particular case, if we dissect, if we take out everything just ahead of the medulla, we just take the cerebrum, we take out the cerebellum, that's it. So if we are taking out the cerebellum, the cerebrum of the fishes or amphibians, but we are not taking out the medulla part, we are not taking out the spinal cord. So what we have is we have medulla after it, and we have the spinal cord. So what we are destroying is the cerebrum and the cerebellum. So what is left in the fish or the amphibian is the medulla and the spinal cord. So what has been seen that there is no, you can say, negative effect on the locomotion. So locomotion in this particular case is not affected. So this is very, very important experiment. So if we take out the cerebrum and the cerebellum, there is no effect on the locomotion of the fish or the amphibian. So that simply gives you an idea that medulla is very, very important part of the brain that controls the postural control and the equilibrium or the locomotory senses. But on the contrary, if I talk about the, suppose I take the birds and the mammals into account for this particular experiment, if I take out the cerebrum and the cerebellum of the birds and the mammals, the medulla is left behind, the spinal cord is left behind, but here in these forms there is no locomotion as such. Locomotion is affected in the case of the birds and the mammals, and they are not like in the condition to locomote like with the medulla only. So by this experiment it is like clear that in the case of the birds and the mammals, the medulla, it has got less control over the locomotion because it is not affected. So it has got the respiratory center, the inspiratory and the expiratory centers. So it is more related to the basic respiratory and the cardiovascular systems in the case of birds and mammals, and it is less related with the locomotion in this particular case. So we are finished with the first part.